Good afternoon, everyone. We'll continue where we left. I think we were discussing about triptopyron. In the last class, I have given you some details about triptopyron, and there are two levels which we will discuss today in little more detail. And then we'll go with some more examples. And towards the end of today's lecture, I would want to give an introduction to the regulation of lambda phage, lysogeny, lytic, and so on. So today we'll only learn components of lysogeny, lytic decision making, but otherwise the focus is on the other regulatory examples of regulation that we have in prokaryotes. So in today's class and in the next class, we will finish prokaryotic gene regulation, and then on Friday and Saturday, hopefully we'll finish. So sorry, we'll finish the eukaryotic gene regulation, which will be a little more, little brief in nature. But we'll also want to know when is the last date? It's on 10th or 9th. Can anybody say?
10th, sir.
Okay. So in that case, I will have 7 and 10, in which classes I would want to review a few things and probably discuss a model paper so that you will have some understanding of it. Okay. Let's plan it that way. Yes. So either way, I already posted you some videos of gene regulation that I have recorded last year. And I think apart from the audio, the content of it is actually good. And maybe, yeah, I guess I'm using the same PPT, and you probably can also go through that in case if you have any questions. And you're always welcome to talk to me, message me, so we can discuss anytime if there is a question. Okay, so what we are talking is I will just quickly help you recollect what is negative regulation. Negative is about repressor. If you're talking about repressor, then it is negative regulation. And the second part is the type of control, right? We are talking about negative is about when we discuss about a repressor. And whether it's inducible or repressible is about the molecule or a ligand that has the influence on the repressor. So some are, I think we already discussed this several times, so we'll skip it. Negative inducible, negative repressible, positive inducible, and positive repressible. So triptopyron we already discussed. Yeah, before we go into in any of these regulation that you are talking about, especially in the eukaryotes, and we talk about an operon, right? And know the structure of it, meaning that where is the promoter and what gene is downstream. You may also know the significance of it. What does the regulated gene, what is the function of the regulated gene, right? Significance. And you will also know whether it is type of control. That is what you should also try to know. Type of control, that is one thing. And yeah, how does it regulation, there are in which conditions it's expressed and in which conditions it is repressed. So if you know this is the way you try to build things. In some cases we should know very good details, like in lacoperon and triptopyron. The others probably you should know the essence of it. Okay, triptopyron, it is a biosynthetic operon. That means tryptophan, these enzymes will produce the tryptophan amino acid. And when does tryptophan need to be produced? When there is lack of supply. If this exogenous, that means outside the cell. Endogenous means that is synthesized from within. So tryptophan, if it is available from the outside, then you don't need to we don't need to make it, isn't it? Whereas if it is there is no supply from outside, then it has to start synthesizing. So the cell should be able to sense if there is if the tryptophan is available to the cell or not. If it is not, then it should activate trypoperon and start producing the genes, start expressing the genes so that the enzymes will form, and then we'll have the production of tryptophan. So for this, there are two levels of regulation. One is at the level of repressor, and I mean at the level of transcriptional initiation, and the second one is at the after the mRNA is being synthesized or while it is being synthesized, right? So when you say about the initiation of transcription, then it is about the repressor binding to the operator. So we already saw this. It's simple and straight. When there is no supply of tryptophan from the outside, the cell signal senses it. And there is one thing, what happens is if there is no tryptophan from supply, then you need the expression of these genes. So typically it is a negative regulon, negative because it's regulated by a repressor. That repressor is inactive when it is not bound by a ligand. You can compare it with the scenario in this case. Okay, 1 second. The graphics tablet is not writing. Let me check. Okay, sorry. Anyway, I'll just move on. So by default, the repressor is inactive. It can only be activated if there is a ligand, and that ligand can be the tryptophan itself. So if the trip repressor cannot bind to the operator because it's in inactive state, therefore RNA polymerase will bind here and will allow the transcription of this operon, leading to the production of the enzymes, which will eventually, through a series of reactions, will produce tryptophan. Right? This operon needs to be active only when there is no supply of external tryptophan. So it can produce enzymes that will synthesize tryptophan. But if there is enough supply, especially from outside, then it doesn't have to produce it. It's a waste of so many enzymes to produce so many enzymes because there is anyway sufficient supply from the outside. So in that case, if tryptophan is abundant, the tryptophan can bind to the repressor, and the repressor is now in an active state, and then it will bind to the operator region. Therefore, there will not be any transcriptional initiation or no transcription. So in this case, the tryptophan operon is repressed. So it is a negative regulon, and it is repressible because the presence of the ligand, that is tryptophan in this case, binds to the repressor and activates it, which will go on bind to it. Otherwise, it is always induced, right? That is how you can take it. So I was talking about attenuation in the last class. I hope you can think of it. There is a later peptide which has a small, which has a small open reading frame, and there are two trip codons within that. And there is also another way of how you see it. The trip E is this first structural protein that is produced, and there are a series of other genes as well. The leader peptide is ahead. It's like something like similar to that of upstream open reading frame. It's not called that way, but just you can think of it. So the mRNA can also has four regions: region 1, 2, 3, and 4. Right? So these regions are complementary in nature, which means that they can base pair. 1 and 2 can base pair. as shown in this case, 1 and 2 can base pair, and 3 and 4 can also base pair. Okay, so this can happen only when we this base pairing can be altered by whether region 1 and 2 are free or not. Okay, say for example, if a ribosome is coming here and there is no tryptophan in this cell, and in that case you will not have trip charged RNA will not be there, then the ribosome that has come here will stall. By being here, as shown in this case, when there is as shown in B in this case, the ribosome is halted there, because of which one cannot base pair with two, but there and so two will base pair with three region. Okay, and in that case the transcription will continue and make the complete mRNA. But if there is high tryptophan and initiation has already started, and ribosome in that case will cross over the first region, even though there are these two trip codons, because there is abundant supply of trip charged tRNA. So ribosome will not pause or stall there. Instead, it will move to the second region also. So 1 and 2 cannot base pair, so 3 and 4 will base pair like this. And in that situation, if 3 and 4 are base paired, you will have transcriptional termination. So you should understand it is a little bit complicated in the sense that when transcription is happening, somehow I'm having some problem with the graphics tablet. I hope it will be settled soon, later maybe. So you have the RNA polymerase is transcribing, and assume that the first part of it has been moved. So region 1 is this one, say. And in prokaryotes, transcription and translation are coupled, something like this. Say this is a ribosome that I have drawn. And while transcription is happening, translation is also happening in this direction. So if there is no if the tryptophan is less, then the ribosome will move very slowly or get stalled on region 1. In that case, loop 2 and 3 will form, and there is no hindrance for that. Structure does not hinder the RNA polymerase from transcribing further. Whereas if there is abundant tryptophan, the ribosome would have moved to different location like this. In that situation, a structure like this 3 and 4 forms. Okay, you please just hang on. I should have to correct my graphics tablet. It is bothering a little bit. Remind me where I was. So there is some okay. Okay, there is an issue. I cannot change it. Okay, let's just go ahead. Okay, you can see it, right? Somebody, can you say it, please?
Yes, sir.
Okay. So I hope you understood the essence of it, in the sense that it is, if 3 and 4 form a structure like in this case, 3 and 4 form hairpin loop, then it will disrupt the interactions or disrupt the RNA polymerase that is actually transcribing the trypoperon. But so you can think of it like this: 3 and 4 hairpin, a hairpin formed because of 3 and 4. You can think of it as inhibitory hairpin loop, inhibiting the RNA polymerase that is actually transcribing at that location. So anytime this is the RNA polymerase, you have ribosome, and if 3 and 4 forms, the RNA polymerase falls off. You can see in situation in A, when high tryptophan is there, or when there is no protein synthesis also. When there is no protein synthesis, transcription has happened. So is happening, initiation has happened, and there is no ribosome to halt the base pairing of formation of hairpin between 1 and 2. If 1 and 2 form, then 3 and 4 will also form. 3 and 4 is a kind of inhibitory hairpin loop. Because of its structure, it disrupts the RNA polymerase activity and transcription is terminated. Okay, remember it that way. So it is a kind of repetition of what I already said. You have tryptophan. The hairpin loops form something like this. When tryptophan is high, you will have 3 and 4 base pairing, and there is a poly-U stretch that is there, that is present downstream of the 3 and 4 region within the leader region itself, and that this structure terminates transcription. Whereas if tryptophan is abundant, one is when tryptophan is low, then ribosome stalls here, something like this, because there are tryptophan codons are there. So one is not able to base pair, so 2 and 3 will base pair like this, and so 3-4 hairpin loop is obstructed. So transcription continues. So there are about how it disrupts the transcription or influences the RNA polymerase. We'll have to go into a lot of structural details. But for the time being, what you should know is the presence or absence of tryptophan will influence the ribosome, the ability of ribosome to read the leader region. That is, the ability of ribosome to read the leader region will disrupt the hairpin structures that are going to form. And these hairpin structures are going to influence the RNA polymerase, whether transcription should continue or should be terminated. So that's a complex level of regulation. You see, for tryptophan, this is an important question also that you have to find yourselves. Why should there be two levels of transcriptional regulation? One is you have with the negative regulation because of trip repressor, and the other one is by attenuation. Why do we need two levels? That's an interesting question. You have to try to read and find out. The textbook itself is sufficient for it, actually. Okay, I'll now move on to the sigma factors, alternative sigma factors. We discussed this a little bit when we are talking about transcription initiation, and there we spoke about sigma factors. In E. coli and many, sigma 70 is the prominent or predominant sigma factor that allows the transcription of most of the genes. But there are certain operons which need to be expressed only during heat shock. What is a heat shock? Typically, if you take something like E. coli that grows in our tummy, our average temperature is around 37 degrees centigrade. So they grow good. That is the optimal temperature for E. coli, Escherichia coli. It's a mesophile in the sense it grows not in the extremes but in normal. So if it goes below 30 degrees or even 20 degrees or so, we call it as cold shock. And if it grows above 42 or 45, okay, then 45 degrees centigrade, then we call it as heat shock. So the protein, you know about protein conformation. They have every protein has an optimal, the structure of the important is everything. Structure and the activity or the ionization of the side groups is important. And that side groups will again influence the structure. So everything is about structure of the protein here. If you change a little bit of chemistry, if you change the energy levels around, then the proteins will change their structures. And these structures could be reversible or irreversible structural changes. Okay. In that case, if I take a protein that is stable at 37 degrees centigrade and I keep it at 30 degrees centigrade, maybe it will lose some of its activity. Or if you even increase the temperature, then also it may affect the structure of the protein, and because of which the activity of the protein is going to get influenced, right? So a bacterium is successful if only it can tolerate these changes in the environment, changes in temperature, pH, or availability of resources, and not, and so on. And the existence of that bacteria is about all the proteins that are present, whether it has the DNA or not, and whether the boundaries are clearly defined or not. These are some of the important things that it should be. So if you take a bacterium that is stable at 37 degrees centigrade and increase it to say 45 degrees centigrade, then some of the proteins are going to be unhappy in the sense that they lose their structural integrity, because of which they will lose their activity. And then the cell is failing to live or exist. And the only other way is, if you fail to live, you're starting to degrade or deteriorate, and that is death. So in animals sorry, in bacterial cells, there are certain ways of how to tolerate these things, and there is one of the sigma factors is called sigma 32, that produces, say, now suppose this is at 37 degrees centigrade, you put it to 42 degrees centigrade, and we have a problem because some of the cells are unfolded or misfolded, right? What should we do? Because of the change in the temperature, the protein structure is affected. What should we do then? We should get rid of these unfolded or misfolded proteins. If you want to get rid of unfolded or misfolded, there are two ways of doing it. One is to fold them properly, fold them back, right? If something has got unfolded, you try to fold it back into correct conformation. The second approach is, if that folding back is not happening, then degrade it. It's unnecessary resources, it's occupying space, it has used up the chemical resources. So you have two strategies: fold them back into proper conformation or degrade. So for degradation, you would require proteases, and for folding them back into proper shape, you need chaperones. Right? Chaperones are other proteins which will allow proper folding of different various proteins. They create an environment where the protein folds up itself into proper structure or conformation. But this is a general talk in that sense, right? You can see how bacterial strategies, it's all evolution has brought them up. Again, it's like whatever is working is retained, whatever doesn't work will be eliminated. And these are some of the strategies. Like, if you have misfolded proteins, try to fold them back. That is the logical solution, because it will conserve the energy from degrading and producing again. It's the simplest and shortest, but that's not always possible. In that case, we need to degrade these proteins so that we will reuse those amino acids to make something better proteins or stress related proteins. Okay, I hope you understood that. And this is the in similar ways we will talk about maybe nitrogen metabolism also has its own sigma 54. If there is no supply of nitrogen is important, like CHNOP, right? In N, nitrogen, almost all, say, proteins and nucleic acids need nitrogen. If that is not in supply, it has proteins for, it has a sigma factor to increase the nitrogen metabolism. So I hope you can see that a bacterial cell typically can have multiple sigma factors, each sigma factor trying to reorient the proteome. What is a proteome? I hope you know these terms. All the genes together in the that are present in the chromosome are called as genome. At any given time, you'll have several types of or 100 types of mRNA genes expressed, and at the mRNA level, you call that as transcriptome. Some of them, most of them would result into forming proteins, and the proteins at any given state of a cell is called as proteome. If you see it this way, the genome always, or majority of the time, or to great extent, is constant in all the cells of one organism. There will be minute changes here and there, but otherwise genome is constant. What are variable? The type of mRNA produced, the number of mRNAs produced are different. So transcriptome is dynamic. At the same time, number of mRNAs translated, types of mRNA translated, to the degree of translation, alternative splicing, and many things are also there. So all those proteins at any given state are called as proteome. Okay, so in bacterial cells, you can see that having a sigma factor for each situation is going to make the proteome to handle that stress. When there is envelope stress, sigma E takes over and produces or expresses genes that are responsible for producing proteins that will handle the envelope stress. If there is heat shock, then that will handle, in the sense that, say, for example, there are chaperones and proteases. They have the promoter region, and that promoter you may have that is specific to be recognized by sigma E. Right? All the heat shock related protein will have promoters that are recognized by sigma E. Similarly, there is a partitioning of genes with their the promoter regions that are recognized by these respective sigma factors. Okay, just you can remember it, that there are the conditions, there are genes, different genes have functions in different situations, and these genes or operons are controlled or expressed by different sigma factors. So bacteria, they have multiple sigma factors, each expressed in conditions that are in which the operons, when expressed, will try to alleviate the effects of that stress. Here is one example for bacteriophage, which we'll also have an introduction to today. Um, so there are several genes on the bacteriophage SPO. You'll have several genes. Some genes are called as early genes, some are called as middle genes, and some are called as late genes. They are called so because as soon as the bacteriophage enters into a bacteria, some genes are expressed early, and that's why they call them as early genes. And some genes are expressed after a while after infection, the bacteriophage entering into the bacterial cell. Then those are called as middle genes, and those that are expressed at the late are referred to as the late genes. How is this order brought about? Why not all the genes express at the same time? Or why should they should always follow this early, then middle, and then late? The key is here. There is one sigma factor that transcribes the first promoter. This sigma factor cannot act on the operons or genes or the promoters of middle genes, right? Or it cannot act on the other promoters as well. So what happens in this case is the architecture, genetic architecture. When you say that, it means that here you have sigma 28, which produces a sigma factor that recognizes the middle genes. The gene of sigma 28 is one of the early genes. That means if these middle genes should be expressed, it only happens after the early genes are expressed, right? And if you see sigma 34, that is a gene of the middle genes. That means it will only be expressed if 28 is expressed, right? It requires 28 to be expressed. That means 28 should have been expressed here. That means it will only happen after transcription of this. That is how the order is made. So 28 as a part of early genes is expressed, which will then start the expression of 34 or the middle genes, of which is a sigma 34 gene, which will then be after upon production will then activate the transcription of late genes. You can see that there is no other order that is possible. It's only early, middle, and late. Never the other way, because their sigma factors are under the control, a serial control like this, right? That's a very smart way of doing stuff. So here are some more, a few examples. I will try to be brief in these, but otherwise they're specific for it. It's not global regulation. If you talk about sigma factors, they are they change the whole proteome, right? They are highly specific, and they control multiple genes. At the same time, they can change the proteome of the bacterium so that it can withstand the stress that is coming about. Here we are, some of them are specific or gene specific regulations that we are talking about. One of it is NTRC. It's involved in gene of nitrogen metabolism, which produces GLN-A or glutamine synthetase. So NTRC has a activator binding protein. When there is another signal molecule. Anytime there is the nitrogen is not high in concentration, that gets activated, which will then activate NTRC, or NTRC will bind to the NTRC or the activator binding region, and then NTRC will recruit sigma 54 along with RNA polymerase and allow the expression of GLN-A. And that will produce several, increase the nitrogen assimilation, right? Glutamine synthetase. And there are many other things which we will not discuss, okay? But you can think of NTRC as an activator that recruits RNA polymerase with a specific sigma factor, sigma 54, to the GLN-A gene or operon. This is something interesting. When there is mercury stress, you need to produce certain protein, a gene, say MERT, which will handle the stress of mercury. Mercury is toxic to the cells. The regulation is interesting because it is not like the way we think of it. There are some things you need to think critically about it. So typically we want, we have -10 and minus, okay, it's not writing in that location, so I'll write here -10 and -35. The green one that is showed here, okay, -10 and -35. In C, you can see that there is a certain way of orientation, and the distance between -10 and -35 is typically between 15 to 17 base pairs. The spacer is also important. I hope you remember that. If you see the MER operon, or the -10 and -35, the distance is 19 base pairs, right? It is more than, it is unusual spacing. So if you keep following this line that I say, here is that, and here, and from there on this graphics tablet is not writing, so you adjust and follow the green. If I can write it linearly, I would want -10. I mean, you can assume -10 and -35. Even if they are folded upon, I would want that they both are on the same surface, so that the sigma factor can interact both of these in a proper orientation. Instead, If the situation is little bit that the sequence of minus 10 here is little bent towards the inside, then the sigma factor cannot interact with minus 10 and minus 35 efficiently at the same time, right? And that is happening because this is 2 or 3 bases more longer than it should be. It should be between 15 to 17 is typically a good spacing between minus 10 and minus 35. But in MER-T promoter, that is upstream of MER-T, it is little longer. So there is a protein, a regulator, we'll call it as a regulator, MER-R, and it can change its conformation from active to inactive, or in both the cases, it binds to the DNA. If it binds to, if it is bound to DNA, MER-R in its native conformation, we'll call that as native conformation. The minus 10 region is on the opposite side, almost opposite side. So the sigma factor cannot interact with minus 10 and minus 35 at the same time. So because of which there is no transcriptional initiation and transcription does not happen. Transcription of MER-T does not happen. But if mercury is present in high concentration, then MER-R, it binds to MER-R and changes its conformation. Now you can call it as an altered or probably a different conformation. And when it changes its conformation, it twists the DNA a little bit like this. So you notice only this part. Minus 10 is oriented slightly outwards now in comparison to that of minus 35. But because MER-R changed its conformation and it twisted the DNA a little bit, now the minus 10 is in proper orientation. Now sigma factor can interact with minus 10 and also minus 35 at the same time, and transcription of MER-T can happen. Isn't it wonderful? It's not like there is a design in this. It's like anything is possible and whatever works will be retained. That's evolution. Anything is possible. When I said it is, there are a lot of variations that will happen spontaneously, undirected changes or variations will occur, and selection is kind of directed, and the direction is whether it works or not. If it works, and what do you mean by whether it works or not? That means is it helping the bacterium or not in this case. So a complex, you can find it this way. See, it's simple. MER-R is binding to DNA whether mercury is present or not. But in the change is just by the presence of mercury or not, MER-T is expressed or not. It depends, right? The expression is dependent then on the presence of mercury. And that's a nice thing. Why I'm saying ingenious and all these things many a times I use, the word is, or my intent is that I appreciate or I wonder the intelligence of those people who could find it. It's not one person who finds it. These are difficult things. We don't know how things happen. Keeping an open mind is very important, because who would have thought that this is how it might happen? These are probably some models. And I think they made a crystal structure of MER-R with mercury and without mercury along with the DNA, and then they couldn't find the structural changes, and that is when they propose it. So the logic or the reasoning capacity or the imagination of scientists is what wonders me more, because it's not easy to find out. So try to read biology more in a way to improve your brain power, the ability, the openness of your brain, and also to logically reason and still be open that maybe there is something more that I cannot think of it yet. That's how we should approach in science. Let's move ahead. So there is one another operon that is called for arabinose. It is similar to that of lac operon in the sense it is a metabolic operon. That means, like in lactose, we have lac operon, which will metabolize lactose. The same is the case with arabinose operon, that is to metabolize arabinose. The genes present in the arabinose operon are synthesized, which will degrade arabinose to produce other compounds or energy. So, arabinose, there is one part that I'm not discussing, that is with CAP, catabolite activator protein, that is also involved in this case, but I will only stick to arabinose, regulation of arabinose by ARA-C. ARA-C is kind of activator or a repressor. It has a dual role in a way. You can also think of the previous one that we have discussed about MER-R. It is an activator or a repressor. In the sense, MER-R in that position, if it is in this case, it is kind of repressor because it will stabilize this kind of arrangement, because of which RNA polymerase cannot bind here. But if it is activated, then it is now acting as an activator. By twisting, it is allowing the RNA polymerase to bind. ARA-C is also in a similar situation or has similar properties. So there, first we'll learn about the structure. Here is the pBAD, this promoter, ARA-P. And then there are certain operator regions or regulator regions. We'll call it as regulator regions because operator regions, usually we talk about binding of a repressor. But in this case, ARA-C can act as a repressor or activator based depending on the ligand that is bound. The ligand that binds here is arabinose itself. So first thing is, if arabinose is present, we want the production of metabolic genes that will degrade arabinose to produce energy or compounds that might be utilized through glycolysis or other processes. And so, yeah, let me finish also this one. Arabinose is ARA-C is a repressor or activator depending on arabinose. So the region that it binds, we cannot truly call it as operator region because operator region we usually associate it with binding of repressor. So there are operator regions here, ARA-I, and this is the operator region ARA-O. So when arabinose is not present in the cell, we don't want the expression of it. In that case, ARA-C has a different conformation. So ARA-C has one conformation, and when it is bound, you can think of it as a different conformation. There are conformational changes, and that is whether the ligand is bound or not. When it is unbound with the ligase, it has a different conformation, because of which it forms a dimer. And as represented here, it is forming a dimer based on a different structure, the side part of it, like this. And it can interact with this part. So the ARA-O2, operator 2, and ARA-1I, 1, you can call it as I1, sorry. So in this way, when it binds, it will prevent the RNA polymerase from binding and initiating transcription. So it is acting as a repressor in this case. If arabinose is bound to ARA-C, it has a different conformation, and it forms a dimer also, but it forms a dimer in a different way. Previously, you see that these two interacting are the points, but here now, because of the change in conformation, you have a different points of contact, and the dimerization is different. And in this conformation, it will influence or activate or allow or promote the binding of RNA polymerase and initiation of transcription. So in this case, it is acting as an activator. Okay, quickly, let's see. When glucose is present, what do we want? We when like we spoke about lac operon, right? When glucose is present, we don't need lac genes to be expressed, because glucose is a better metabolite that produces ready energy. So the same way, when glucose is present, we don't want arabinose operon to be expressed. So CAP is involved in that to repress arabinose operon, which I am not discussing now. You can have a look at the textbook. But when arabinose is present and glucose is absent, then you will have this situation where we have an expression of this. When you have both glucose and arabinose, still glucose is a better opportunity. In that case too, you will have arabinose not expressed. That has to do with the cap protein that we are not discussing. Okay, but I hope you understood how one protein can act as an activator or a repressor based on the ligand that it binds to and the mechanisms of activation and transcription that can happen. So this is, I will move on. So I have 5 minutes. I will give a quick introduction to the lambda phages, and in the next class, I will discuss about the regulation of this one. So bacteriophages are viruses that infect bacteria, and when they infect, they attach to the surface of the bacterium. The protein coat is left out typically, and the genome is inserted into the bacterial cell. And now there are two situations. There are some bacteriophages can undergo, especially if we talk about an lambda phage. Many bacteriophages just undergo lytic phases. You I mean, the bacteriophage injects the DNA, it will multiply, lyse the cell, and then they are released out. These will again go and infect more and will release more. That is typical reproductive cycle continuously happening. When you see, there is another that is called as lytic cycle, because there is lysis of the host is getting lysed. There is another one called as lysogenic cycle, and this is smarter in a way. What is the purpose? There is no purpose for bacteria as a conscious purpose, but what happens is reproduction. If something is not reproducing, it will go extinct. Any species in which reproduction is not there, then it will go extinct, right? If it is not producing in contextually healthy offspring, it will go extinct. That is how genes and genomes with good and better genes will survive. Better groups of genes as genomes will survive, and the others will die out, right? Why lysogeny is an interesting scenario, where the DNA that is inserted gets incorporated into the genome, bacterial genome, and this is called as prophage. Phage genome that is inserted into the bacterial genome. I hope you recollect that when we discussed about site-specific recombination, we discussed about at P and at B and so on, the molecular nature of how this site-specific recombination happens. Int protein integrase, excisase, IHF, integration host factor. We discussed some of these things. I hope you remember how insertion and deletion can happen. In that example, we discussed that. Here we are seeing a different, the regulation of that insertion and deletion, right? So because it has direct implications. In this case, when it gets incorporated and bacteria divides along with it, the phage also divides. What is the problem? It is two-way good thing. Say, for example, in this situation, in lytic cycle alone, the phage will win, right? Phage wins, bacteria dies, right? Why is that? Because bacteria is dying out, and phages, you have multiple numbers. Eventually, if there are no bacteria left at all, what will happen to the phages? They need something to infect and reproduce, and they are not there. So that is also going to go extinct, right? Lysogeny, I think, is an evolved strategy, that in which case you see in the lysogeny, the bacterium is trying to reproduce, phage is trying to reproduce. The only problem is that phage is dependent on bacteria for reproduction. Bacteria by itself is going to reproduce taking nutrients from the outside. Phage is a parasite on bacteria. And we have situation here where bacterial genome and phage genome have become one genome. And by doing so, they are both are succeeding, right? Bacteria is alive and phage is also alive. Do you remember sauce response, where we said when there is extensive UV damage, then what happens in that case? When there is extensive UV damage, you will have a lot of bacteria that is, 1 second, where are you people? Yeah. When there is extensive DNA damage, is it good for the bacterium to survive, or it's not good for the bacterium to survive? Sorry, it's bad for the bacterial cell to survive. Bacteria is going to die. In that case, what about the prophage? The prophage is also going to die if the bacteria dies like this. Virus is only stable outside in the environment if only it has the capsid and other components that are required. If they are not there, if you take out the phage genome and put outside, it's going to go, it's no use, there is no life in it, it will not give rise to more. So in that case, the source response, what lambda phage does, from lysogeny, it will induce lytic cycle. It's like somebody thinking this house is burning out. Let's pack our bag and leave to a different house. It's something like that. So these bacteria that can undergo lysogeny, or phages that can undergo lysogeny are called as temperate phages. Okay, so I think that is sufficient introduction to it. So the question what we are going to read about in this particular regulation is, if a phage has introduced its genome into a bacterium, should it take lysogenic pathway or should it take lytic pathway? How does a phage decide what to do? And that is what we are going to read about. I encourage you to watch the videos from the last year that I have sent. Please watch it so that we can finish it in one class. Otherwise, it might take long. I don't think I will spend more time than that. I want to finish in the next class the regulation of this lysogeny lytic decision making by lambda phage, and also translational regulation in bacteria. Okay, please have a look at it. It's actually interesting, and there are too many details. I think we won't ask you in the exam for so many details, but the overall, it's interesting, and also probably you can draw inspiration by trying to read about these finer details that will empower you to understand complex things, and that is what we have to deal with, and that's how we grow our cognitive skills. 1 second, we stay online.